È la sessione dedicata al premio Rita Levi Montalcini. È una sessione dove lo scorso anno abbiamo iniziato un percorso che vorremmo continuare anche nei prossimi anni, di far precedere la consegna del premio Rita Levi Montalcini da una lettura magistrale che abbiamo definito Rita Levi Montalcini Prize Lecture, e mi viene in mente, prima di presentarvi chi terrà questa lettura magistrale, mi viene in mente una domanda che ieri in un'intervista televisiva mi hanno fatto: perché questo premio Rita Levi Montalcini, cosa significa per me, che tutti gli anni sono finché potrò qui. Ovviamente prima c'era Rita Levi Montalcini, adesso indegnamente ci sono io a consegnare questo premio. E una delle cose che ho risposto è che noi abbiamo seguito come Fondazione il percorso di tanti ricercatori che hanno cominciato con noi e hanno proseguito con noi, perché abbiamo anche continuato a finanziare le loro ricerche e abbiamo anche sostenuto il loro percorso quando era necessario per il ritorno in Italia. Il piacere che ho è di presentare la lecture di quest'anno e di chiamare Stefano Pluchino. Dicevo prima quello perché Stefano Pluchino io l'ho conosciuto che era ancora studente all'Università di Siena e ce l'ha presentato proprio una persona della sezione locale dell'associazione e il neurologo che lo seguiva. Il suo percorso è stato lungo. In questo momento è professore di neuroimmunologia rigenerativa e consultant di neurologia al reparto di Clinical Neuroscience all'Università di Cambridge e la sua ricerca di 20 anni l'ha portato in moltissime università come ricordavo prima è stato anche uno dei premiati del premio Rita Levi Montalcini nel 2007. A te il compito di questa lettura che ancora una volta ci porta a vedere un contesto più ampio che non quello di una singola malattia, perché quello che possiamo fare nella ricerca è cambiare la realtà per tutti. A te Stefano e benvenuto in Italia, anche se ogni tanto torni.
Thanks very much, Mario. And I have a few disclosures to make before we get into the real matters of the presentation. The disclosure are that I will be specifically focusing towards progressive MS and human studies. I will be emphasizing and elaborating on the promise of single-cell or single-cell inspired and big data studies, and aiming at putting together the overarching vision of connecting MS with other neurological conditions to identify and target disease-associated cross-concern mechanisms of disease to preserve the neurological reserve of the brain, which is what Giancarlo just highlighted as protecting the brain. And then finally, of course, I have to apologize with those of you with the work which has not been cited or elaborated enough in my presentation, and then we will conclude by celebrating this year FISM Rita Levi Montalcini Award. So we start from the agenda that the National MS Society, but actually it has been extensively endorsed by a number of other funding bodies, has put together earlier this year, which is the agenda for research, the pathways to cure for MS, which distinguishes between different stages and aspects and phenotypes of the disease, prioritizing three major actions. The first of which is stopping MS by developing early detection exercises or precision medicine approaches. Precision medicine is something which you will listen over and over again. The second task is to restore with different levels of expectation from multiple stakeholders. The big picture is that we all want to regenerate the brain and we all want to restore functional activities, but we don't know yet how to make it clinically relevant. And the final task is to, of course, to end MS, including primary prevention, with the expectation of not adding new cases of MS anymore, or still ending by blocking the transition from fully resilient phases of the disease into irreversible phases of the disease. So let's start from a very brief yet very focused introduction to progressive MS, starting from the big picture and going down to single cells. So MS is everything but an easy disease. And in this very recent review article published in Nature Review Neurology a couple of weeks ago, you can see how complex progressive MS is in the brain, with a number of different types of lesions and brain behaviors. Of course, there is some of these lesions which are very, very exciting to study, which are being proposed as potential biomarkers of disease or progression. For example, these chronically active lesions, which are slowly expanding over time. But there is also areas of the brain which seems to be normal-like, areas with excess of homeostatic microglia, which is the microglia out of which we are expecting a reduction of inflammatory activities. There is lesions in the cerebral cortex. There is B-cell aggregates in form of ectopic tertiary lymphoid follicles, which might activate complement-dependent secondary damage. There is lesions with early axonal injury, and there is also areas of the brain with very little remyelination. So it is complex, it is heterogeneous, and it is a big challenge for next-generation medicine. And in this context, I'm a very big fan of animal models. We've been making extensive use of animal disease models or animal MS-like lesion models, from fish to small laboratory animals to non-human primates. But the fact is that these animal models are interesting, they are relevant, they are a toolkit which we can use on demand to mimic or to model specific aspects of the pathobiology of the human disease, but none of this is superior. They're eventually complementary to each other, but none of this is superior. And importantly, we don't have yet a fully translatable animal disease model for progressive MS yet. And importantly, tissue regeneration works beautifully in animal models, but eventually it is much less likely to work in the human brain. And another important confounding factor, which is lifespan or prediction of lifespan and aging, is very difficult to model, to mimic in a small laboratory animal whose life is pretty much 2 or 2.5 years, which is very difficult to extrapolate to the human brain. So having made this introduction, I think it's important to elaborate on the unique opportunity that the community is living when considering that we are facing the age of big data, of human atlases, and of data and resources available. This is the cover page of Science, last issues of Science, where 3 back-to-back papers on this international, worldwide, multi-million initiative, which is called the Human Cell Atlas, were published. And the Human Cell Atlas is a fantastic initiative endorsed by a number of funding bodies, public and private. As you can see, the British Heart Foundation, the Wellcome Trust, the MRC, the NIH, but also the Chan Zuckerberg Initiative and a few other trusts, which holds the ambition to apply single-cell genomics and spatial biology protocols in combination with unsupervised machine learning algorithms to create a cellular reference with the position, function, and characteristics of every cell type in the human body. And the ambition, in more details, is to catalog each cell type in the human body, to map their location in tissues, to identify cell states, to trace the developmental trajectories of each cell, to reveal cell processes involved in development and adulthood and disease, and to provide an openly available, full access, globally representative resource to study and understand health and disease. This is very global, this is available, but what about MS? There is something which is already being applied to the MS question and to the question applied to other neurodegenerative conditions, not as ambitious as the Human Cell Atlas or the Tabula Sapiens, but which is very close. And I have to thank Giuseppe Gazzola, who inspired me on showing you this picture, which is a very nice picture from the sky of New York and Central Park, because Giuseppe, when I was trying to elaborate on the single-cell analysis and the potential that to study old, was making a comparison with a drone. And in fact, by applying single-cell analysis of transcriptional and epigenetic states in the human adult MS brain, we are providing the opportunity to investigate cellular and molecular aspects of very complex yet poorly understood neural-immune interactions in the human brain with unprecedented details. So we start from the sky and we identify different clusters of cells. So you can see there microglia, OPCs, astrocytes, all the relevant cells that we have been studying by candidate approaches in the last 25 years or so, by using unsupervised, unbiased single-cell technology. And we go into details and we identify the individual clusters with spatial resolution and single-cell details in the human postmortem brain. You see that, for example, neurons are indeed pretty much 50% of the cells from the brain, and then a little less represented, you can see astrocytes of different types, endothelial cells, microglia, and OPC. And I've done a lot of work by reviewing all these studies, and here you can find a bullet point preliminary conclusion of what these single-cell analyses are telling us. Basically, we know that in the MS brain, astrocytes are very different from each other. They are very heterogeneous. They are disease-associated, but not yet disease-specific. They undergo senescence, and they are predominantly neurotoxic. So they are bad guys. We also know that OPCs and oligodendrocytes, which have been extensively receiving attention, are heterogeneous. They are stressed in the MS brain. They are profoundly dysregulated transcriptionally and functionally, and the MS brain, specifically in the core of the lesion and in the normal appearing white matter, shows a selective loss of OPC and entire OPC and oligodendrocyte clusters, which was not expected. We also know that neurons are heterogeneous, but they are very vulnerable in the MS brain, and there is a selective loss of neurons residing in layer 2 of the cerebral cortex in those patients showing cortical gray matter pathology in MS. And we also know that microglia is prominent, is heterogeneous. Again, it is disease-associated, but not yet disease-specific, and predominantly neurotoxic. And microglia is a hot topic in neuroimmunology in these days, because the single-cell analysis are also being very much supported by clinical studies. In this case, I'm showing you a PET imaging study performed on people with progressive MS and with gray matter pathology, which shows that microglia and macrophages undergoing activation are diffusely present in the gray matter of people with secondary progressive MS, and that in MS, increase of PET imaging signals suggesting microglia accumulation and activation correlate with disability, impaired cognition, and cortical thinning. And of course, the very, very expected smoldering lesions, which had been described very extensively post-mortem for a number of years, but which Martina Pfinter and others a couple of years ago was able to demonstrate by high-field in vivo 7 Tesla phase MRI to be imaged as well. And in fact, we see that there is a small proportion of lesions in the MS brain, specifically in the brain of people with progressive MS, which shows a very interesting perilesional Rim enhancement, which is predictive of iron accumulation and activation of microglia, which are being proposed as sites of chronically active inflammation, which are associated with more aggressive disease course, leading therefore to higher disability, and which occur also in people being offered disease-modifying therapies, which are, of course, ineffective in progressive MS, and that pathologically account for 30% of the lesions showing and featuring smoldering inflammation, smoldering inflammatory demyelination at the edge, remyelination failure, and axonal damage. So at this stage, the question is whether we have to discuss of lesions or disease when thinking of developing a new approach to the understanding and the treatment and the end of progressive MS. It is not just a semantic issue, but it is a biological question. Smoldering inflammation is a very common feature to several conditions and plays a major role in physiology and disease. It is described to be associated with organ specificity, of course, in a number of metabolic diseases, including obesity, diabetes, cardiovascular diseases. And in the context of being a potential driver for disease, it also participates to the initiation and progression of a number of diseases of the immune system, including rheumatoid arthritis, including lupus, psoriasis, and progressive MS. However, the MS brain is impenetrable to signals coming from the periphery, and therefore the question is whether smoldering MS is the real disease that we want to treat when thinking of progressive MS. And there is lots of evidence in support of this smoldering disease concept. And this evidence shows that in the context of a very complex stage of a disease, which is progressively losing what we call neurological reserve of the brain, we observe that there is early axonal damage occurring even from the very early stages of the disease. Demyelination and bioenergetics go wrong during the course of the disease. Smoldering inflammation activates persistently astrocytes and microglia in the brain. There is also evidence of antibody-mediated damage, which can be antigen-specific or antigen-non-specific for molecular mimicry mechanisms. And importantly, there is also faster aging in the MS brain. So all these are overlapping and confounding factors which make these smoldering lesions being accepted as biomarkers of much more complex underlying subtle disease. So aging is important, and the fact that aging is important is confirmed by the fact that aging may have a profound impact on cellular responses, on organs and systems, and may have important implication for the aging of the brain and for the progressive reduction of the neurological reserve. And the fact that aging is important is also supported by this unprecedented global initiative that was conducted by a huge number of institutes and investigators. It is called Brain Chart for the Human Lifespan. So this is a fantastic study which was published in Nature last week, which ambition is to provide a standardized assessment of brain development during aging in physiology and in the disease. You can see from the numbers the ambition of the study. So they recruited over 100,000 human participants between 115 days of life until 100 years of age, which were subjected to more than 120,000 scans from over 100 primary studies. And these brain charts are showing us what is the behavior of the human brain across life. By comparing males and females, so you see that these brain charts are providing standardized measures of volumes, of areas of the brain. The total cerebral volume, the surface, the cortical thickness, the gray matter, white matter. So this is a reference which is being proposed to be the starting point for future studies whose goal would be to identify new outliers from the brain chart applied to development, disease, physiology. Again, this is a very broad example, not yet applicable in a very straightforward way to MS, but we have also already MS-specific cases. And this is a very, very nice study coming from the MAGNIMS consortium and published by the group led by Olga Ciccarelli last year, where the new algorithm which was developed is called what they call BrainPad. This BrainPad is a brain predicted age difference. So basically this is a subtraction between the individual chronological age and this age predicted by a completely unsupervised machine learning algorithm on voxel-wise neuroimaging data. Comparing people with clinically isolated syndrome, people with relapsing remitting MS, primary, secondary, progressive MS, and aiming at assessing whether MS is associated with a higher apparent brain age and whether that apparent brain age has implications for development of disability, response to therapies, and brain physiology. And it is very exciting to realize how faithful is the accuracy of this brain pad, and the fact that people with MS have much higher brain pad compared to controls, that there is a relationship between the predicted age by machine learning and disability already at the study entry, that the brain pad increases over time, and the brain pad at the baseline is also a good predictor of future disability progression and response to treatment. And importantly, you can see here how people with primary progressive MS already at the study entry have a predicted brain age, which is 11 years old, higher than control. So the brain of people with MS is older and gets older with a much faster pace as compared to controls. So aging is important also because aging might further impair what we call and what several disciplines in neurology, specifically focusing on neurodegenerative conditions, call brain resilience or brain resistance to the disease. The higher the age, the higher the brain pad, the more fragile the disease. And in fact, MS is a disease which is associated over decades of life with an increase of years with disability, but not an increase of years with life lost. So it is not a disease which can be defined to be lethal or fatal, but it is a disease which is profoundly characterized by accumulation of chronic irreversible disability in life. And this is a very kind of epidemiological concept, but if we consider aging from a pure biological perspective, and we go deep dive into some of those single cell studies, and the one that I'm quoting here is the Nature paper that Martina Absinthe and Danny Reich published last year. You can see that by dissecting specifically some glial population in the postmortem MS brain, you realize two things. That these cells are not normal at all, that there is significant amount of stress expressed transcriptionally by oligodendrocytes or PCs and astrocytes, and that 47% of the astrocytes in the core of the lesion are senescent, are aged. So the conclusion is logical that, that the reduction of the neurological reserve of the brain very likely begins very early in the disease, even when patients are clinically asymptomatic, and brain volume correlates with age, MS duration, levels of disability, and MRI markers. Okay, so I'm going to give you a couple of case studies on what we do in the lab in Cambridge, which I think fits very well with the general agenda and with the expectation from Paola, and with the ambition of reconsidering a number of key pathobiological aspects from a different angle, eventually developing unorthodox approaches. And the two case studies that I'm sharing with you are about fuels and triggers of the smoldering disease or smoldering inflammation. The first case study is not just inspired or led, but it is continuously driven and readjusted by Luca Peruzzotti I met in the lab, who is fantastic. I wouldn't say he's a friend, he's a scientific mate or buddy, if you like. And this project, the fuel hypothesis, comes from Luca specifically. And the question that Luca tries to address biologically starts from a philosophical quote of Ludwig Feuerbach, saying that basically we are what we eat. So basically, if we look into what we eat and we play with our fuels, we might be reprogrammed. We might change face and shape and behavior. And the question specifically that Luca is trying to address scientifically is whether cell metabolism, mitochondria, and metabolites play a major role in the regulation of pathogenic myeloid cell responses in the persistently inflamed brain. It is a very ambitious question, and I'll show you a few examples of how Luca is addressing this question. So we start from the fact that mitochondria are central hubs for cellular responses, And these hubs affect main cell functions in nature, with a specific focus in the lab towards how mitochondria can regulate myeloid responses. We know that myeloid cells are very, very versatile. They fluctuate between different functional states very easily and very rapidly. They can change rapidly from their quiescence towards a type 1 activation, which is pro-inflammatory, or towards a type 2 activation, which is anti-inflammatory or pro-angiogenic. And mitochondria play a major role. And in this context, mitochondria and metabolism are playing a role as metabolic hubs, where succinate, which is highlighted in the picture, plays a major role because it is able to induce IL-1 beta through IF-1 alpha in cells. Complex 1 drives the production of reactive oxygen species via reverse electron transport. The TCA cycle becomes disrupted and it is repurposed from classical energy production to production of cytokines and reactive oxygen species. And the consequence of this, when all these mediators of a TCA cycle being repurposed are being secreted, is that neurotoxic damage is observed. On the other end, mitochondria can also work as signaling hubs, where again succinate is released extracellularly, and it signals via SACNAR1, which is its cognate receptor, onto SACNAR1 expressing cells, most of which are inflammatory cells. So succinate is a potential major player of the perpetuation of chronic inflammation into otherwise inaccessible organs or sites. And in this case, mitochondria are triggered and continuously fueled by extracellular succinate. And in fact, SACNAR1 signaling in immune cells is important to perpetuate inflammation. In this context, what Luca started doing, together with Corey Willis and Greg Zark in the lab, was to develop an ontology and temporal dynamic approach to myeloid cells in mice with MOG-induced experimental autoimmune encephalomyelitis. We are making good use of this double fluorescent reporter gene mouse, which allows you a perfect distinction between CNS infiltrating blood-derived macrophages, which are red fluorescent protein identifiable, and YFP, and RFP CNS resident locally recruited microglia. So we can distinguish by FACS analysis between macrophages coming from the blood and microglia being recruited locally in the brain. And the first experiment that Luca and collaborators did was to do an ex vivo single cell RNA-seq experiment, distinguish between controls, peak of the disease, and chronic disease in EA. So without going into details, you can already appreciate that microglia is not present in controls, which is not surprising, but becomes a very predominant part of the myeloid cell infiltrate in the chronic phase post-disease induction, while the acute phase is equally dominated by macrophages and microglia as well. But there is no much macrophages chronically to EA induction. And then the second experiment was an unsupervised cluster analysis, distinguishing between homeostatic, disease-associated, or damage-associated subsets of myeloid cells. And again, take-home message is that we do see two major clusters. One is the one in red, which is cluster 4, which codes for disease-associated microglia. And the other one is the blue one, number 1, which is homeostatic microglia. And you see that homeostatic microglia disappears at peak of the disease, but appears again chronically to the disease induction, while the great majority of the disease-associated microglia are found chronically after disease induction rather than acutely. This is not yet fully informative, but by applying, again, another level of unsupervised analysis to cluster 4, which is the microglia chronic cluster, Luca was able to identify 3 subclusters, 2 of which are disease-associated, transcriptionally speaking. Another one is kind of masked into this disease-associated microglia, but transcriptionally reminiscent of homeostatic microglia. And by specifically focusing at subclusters 4.1 and 4.3, which are the 2 pathogenic or disease-associated clusters, we find a selective upregulation of GO terms coding for forward and reverse electron transport, which are those mechanisms regulating mitochondrial complex 1 mediated production and release of neurotoxic factors, including reactive oxygen species. So at this stage of characterization, Luca went, again, even deeper, specifically focusing, this is pretty much biased, focusing towards genes coding for glycolysis and OXPHOS, which are two metabolic states that in myeloid cells anticipate type 1 classically activated pro-inflammatory activation for glycolysis and type 2 alternatively activated anti-inflammatory activation for OXPHOS. And to cut a long story short, Luca found a specific differential expression of genes in cluster 4, again, disease-associated microglia, only a chronic disease phase, which was also confirmed by an ex vivo quantification of complex 1 and complex 2 activity out of the brain of these mice with experimental autoimmune encephalomyelitis, showing that microglia chronic only upregulates complex 1 genes and shows an increase in complex 1 activity. So at this stage, more questions. So the fuel sub-hypothesis that Luca and other people in the lab have developed is whether manipulating metabolic pathways responsible for this fluctuation in phenotype and function and potentially neurotoxicity of myeloid progenitor cells is a good strategy to reduce the impact and the extent of smoldering inflammation in models of progressive MS. And of course, we have available in the lab a number of beautiful technologies, including advanced therapeutics, which we have been working for several years, including exosomes, including also conditional gene knockout models. So we start from the most fancy technology on the market now, which is exosomes. There are new stuff for targeted delivery. You can use exosomes to deliver different variety of cargos and payloads from nucleic acid, protein, metabolites, etc. But specifically in our experimental work, which has been very generally supported over the years by the ERC and then by FISM, we have shown that stem cell-derived exosomes are able to traffic functional cytokines or protein receptors to mediate perpetuation of signaling horizontally between cells, and also that they can also function, once released, as a fully independent metabolic units. But in this specific context, which is how to target metabolism in myeloid progenitor cells, what Luca did was to make a discovery by identifying, in the context of a very heterogeneous yet poorly characterized EV fraction coming from stem cells, a subfraction of structurally intact, fully functional, breathable, membrane-surrounded extracellular mitochondria that he called mito-EVs. And these mito-EVs are able to breed when provided with substrates for mitochondrial complexes, and importantly, when they are transferred onto myeloid progenitor cells with disrupted mitochondrial function, they are able to rescue their mitochondrial function in vitro, but more importantly, in vivo in mice with EAE, showing a therapeutic impact which is superimposable to that of parental stem cell therapeutics. The second experiment that Luca and collaborators did, specifically Corey and Greg in the lab, was to focus on SACNA1, which is the cognate receptor for succinate released by type 1 classically activated macrophages in vivo, maybe potentially responsible for the perpetuation of smoldering signals, identifying a specific upregulation and very strong expression of SACNA1 by astrocytes and microglia at the lesion ring. So the question here was, what's the role of the succinate SACNA1 signaling in the perpetuation of smoldering inflammation mediated specifically by astrocytes and microglia? And the first experiment was to look at how SACNA1 loss of function knockout microglia behaves in vivo when activated with type 1 or type 2 like stimuli, observing that basically they are much less type 1 and they are significantly more type 2 when responding to LPS and IL-4 respectively. But interestingly enough, when moving in vivo using a conditional deletion of SACNA1 specifically in microglia in the context of EAE, the interruption of this crosstalk between astrocytes and microglia mediated by succinate SACNA1 interaction leads to much lower disease severity and accumulation of EAE-associated disabilities. Another example that we are very excited about is that going back to the complex 1 activity in the regulation of myeloid responses, we have the opportunity together with Mike Murphy which is working next door at the MSc biology unit to access these NDU-FS4 knockout mice. Actually, they are flox mice, where NDU-FS4 is a nuclear-encoded subunit of the mitochondrial membrane respiratory complex coding for complex 1 activity. So basically, by knocking down NDU-FS4 in a specific cell type, you generate a complex 1 loss of function response. So we moved again to EAE. We induced a complex 1 inactivation chronically after disease induction, and what Luca found was, very interestingly, that this specific gene manipulation led to significant reduced mitochondrial ROS secretion in vivo, to much lower oxidative stress, much lower axonal injury, rescue of clinical symptoms associated with EA, and interestingly enough, a rescue of succinate levels in the CSF to physiological levels. And succinate in the CSF is a very important connection between this experiment and the one that I'm going to show you in a moment. An experiment which comes from our long-standing interest towards advanced therapeutics and stem cell medicines, where Luca basically identified a licensable phenotype in stem cells of the brain. Succinate, in fact, is able to induce a reactive behavior in neurostem cells via SUCNAR1 interaction, whereby neurostem cells are able to respond to extracellular succinate and to scavenge succinate out of the extracellular space. So they are scavengers of this succinate. And this scavenging effect is a major effect in vitro on type 1 classically activated macrophages, which become, in the absence of that excess of extracellular succinate, anti-inflammatory. And of course, it has also very important relevance in vivo when advanced cell therapeutics are applied to animal models of MS by inducing a reprogramming of type 1 classically activated MP responses into type 2 anti-inflammatory and scavenging succinate from the CSF. Okay? So all this is what we've done so far. It's very, very coherent with some very much earlier work that eventually led to the Rita Levi Montalcini Prize that Mario was mentioning back in Naples in 2007. But it is nice to see from this movie, and this movie is actually summarizing over 25 years of work in a few minutes. It is very nice to see that the news and views that Larry Steinman wrote in support of our work back in 2003 with a subtitle that was Collateral Damage Can Be Fixed by Advanced Therapeutics is still very applicable to a context like progressive MS where the target has to be collateral damage. And the homeostatic cell competition with endogenous cells takes very much inspiration not only from stem cells releasing protective factors in vivo, therefore promoting tissue protection, restoration of functions, but also to the succinate thing, which is the first time ever evidence of a stem cells able to take something out, to work as a naturally occurring wasting clearance device is injected through the appropriate biological route into a mouse model of a very complex human disease. The usual argument has always been, okay, beautiful, but what about the old expected mechanism of using stem cells to regenerate the brain, to replace specific parts of the brain which undergo damage and which are being lost over time. This is indeed actually a very small proportion of cells behaving this way in the great majority of our experiments, but also experiments coming from peers and competitors. But I can tell you that there is exceptions. So in this case, we have identified two GFP-labeled exogenous transplanted cells which have migrated towards the corpus callosum of a mouse with EAE, and they've been found very close to a couple of areas which I have highlighted by the dashed line, where the staining for myelin is lost. So these are likely to be areas of demyelination in the corpus callosum of the brain of these mice with EA, and the two exogenous cells are expressing OLIC2, which is a transcription factor that anticipates their progression towards oligodendroglial fate and eventually maturation into malinforming cells. And in fact, by specifically changing a couple of things in the experiments and making an adjustment to our experiments, in collaboration with Robin Franklin, we transplanted stem cells of the brain into laboratory animals that we lesioned with lysolecithin in the dorsal column of the spinal cord, recreating an MS-like lesion. It is very artifactual, but it is accepted by the community as to be an MS-like lesion devoid of adaptive immune responses with plenty of macrophages, but demyelinated. However, rodents are fully proficient in remyelinating the lesion, so we needed to add an additional level of complexity by which the lysolecithin experiment was done in olig-1 knockout mice, which have delayed remyelination, endogenous remyelination, in order to avoid competition between the exogenous cells and the endogenous pool of OPCs. And in this case, as you can see, neural stem cells, which in general are very, very reluctant to become glia or to become oligodendrocytes, instead were able to drive successful and efficient remyelination in vivo. So the environment, together with transcriptional programs, set the stage and set the pace for the behavior of these advanced therapeutics, which still we are not able to predict with full confidence from remotely. So it looks like time is now to develop clinical programs. And in fact, with Valentina Fossati and Frank Lenofer, we started developing control and patient-specific directly induced neural stem cells with the idea of developing personalized autologous stem cell medicine for people with progressive MS. We were very excited. Reprogramming technology was working very well. There were no difference between controls and patient-specific somatic cells being reprogrammed into stably expandable neural stem cells. And we were very, very, very excited. But then this paper came out. And this paper shows that MS, specifically progressive MS, shows in vivo and in vitro, and I'm coming to that in a moment, some significant features of cellular senescence, which might be responsible for the failure of remyelination, like the single-cell sequencing data that I quoted you before. And the first figures that this paper was showing was basically detection of a small pool of SOX2 expressing early-stage stem cells or glial progenitor cells in the MS brain, specifically stratified over demyelinating lesions, showing very high levels of expression of P16, which is a marker of cellular aging, and HNGB1, which is secreted but also nuclear alarmin, one of the major factors of senescence and spread of paracrine senescence in tissues. It is not MS specific, but it is senescence specific. And what the first author of the paper was able to do and it is actually also confirmed by independent studies conducted by Richard Nicholas and Roberta Magliozzi between Verona and Imperial College of London, showing that in chronically inactive MS lesions there is a very, very strong prevalence of senescent cells. And what the first author of the paper was able to do was to model the in vivo, in situ behavior that she had described in the postmortem MS brain using iPSC technology. So basically, she reprogrammed somatic cells from patients and controls into stably expandable pluripotent-like induced pluripotent stem cells, observing also with cells in a dish significant upregulation of P16 and P53, and also identifying HNGB1 as a major secreted factor targetable with rapamycin of the observed behavior. So we were a little bit shocked because that would have implied an additional level of investigation into future personalized stem cell medicine for people with MS, but also we were excited this experiment had significantly inspired the last case study that I'm going to show you today, which is our focus not anymore on fuels, but on triggers of disease or disease progression, specifically mentioning paracrine senescence. And this is because Alex got in touch with us, and we were so lucky to be able to attract Alex from Connecticut, where she did her PhD. And we were also, Alex was very good to secure a personal postdoctoral fellowship from ECTRIMS, and to develop in the lab the driver hypothesis. And the driver hypothesis specifically says that disease-associated senescent progenitor cells might promote the spread of pro-inflammatory and anti-regenerative functions via paracrine senescence in the progressive MS brain. This is very, very, even possibly more ambitious than Luca's hypothesis, and I'll show you in a moment how we are trying to address whether this is the case. So we are using a technology which is biologically more gentle than cellular reprogramming, the one that leads to iPSCs. We are applying direct induction or forward programming of somatic cells into stably expandable brain-specific induced stem cells, which has a much lighter effect on the epigenetic memory of the cell. And we are observing that even in induced stem cells, the P16-associated accelerated aging phenotype is present. And it is also associated with high levels of HNGB1 secreted in tissue culture media. And P16 is also associated to P21 and P53. And this is the starting point. So we have developed a very comprehensive interest towards the understanding of senescence and the modeling of this phenotype in a much more complex experimental setting. And with Bristena Ionescu, who's an MRC-DTP PhD student in a lab, we have put together a very exciting omic approach, which this time doesn't look at single genes, but it looks at metabolites. And by specifically focusing towards the endometabolome of NSCs, we have found a beautiful separation between control and MS cell lines. And the detailed analysis of metabolites peaking up or down when comparing control and MS NSCs shows that, there is a complex metabolic rewiring on PMS NSCs. A metabolic rewiring which is also associated with the significantly higher frequency of lipid droplets in cells, which is a biomarker of increased lipid biosynthesis. And another PhD student in the lab is Nati Prasad, again, MRC DTP supported, together with Gabi Balmus, is looking at DNA damage. And you see here from these very nice pictures how more frequent are these nuclear foci of DNA double-strand breaks in PMS, but not in control neural stem cells, which can be even more nicely visualized by COMET assays, showing much broader and much more intense tails of double-strand DNA breaks in PMS, but not control cells, which implies that there is much more DNA damage, but also a much more DNA damage response in MS cell lines compared to controls. All of which is very much in line with the metabolic features of senescent cells, which we are reading about, including markers of senescence, cell cycle regulators mostly, increased mitochondrial dysfunction, senescence-associated secretory programs, lipid droplets, lipid biosynthesis, metabolic dysfunction. And then with Isabel Berman at the NIA, we also conducted very extensive single-cell analysis on control and MS cell lines, which is beautiful, but which at the moment doesn't tell you anything. But you see that we have identified already a couple of cell clusters which are specifically showing up in PMS, but not in control neural stem cells, which we are proposing as unique disease-associated clusters. And then the most exciting part of the work, which is what we are developing together with Madeleine Lancaster at the LMB in Cambridge. Madeleine is a pioneer in the development of mini brains technology, which she has slightly refined by implementing the 3D very complex architecture of mini brains with these air-liquid interface cerebral organoids, organotypic slice cultures, which are called alico slices, to model fetal neocortical development in a dish. In this context, after extensive discussions that Alex and I and Madeleine had under the umbrella of the Cambridge Centre for Myelin Repair, which is funded by the UK MS Society, we proposed to model the impact of paracrine senescence on brain fitness and longevity with human hybrids, creating hybrids between the exogenous NSCs from patients or controls and the otherwise developmentally mature, non-senescent, completely fit, completely resilient brain organoid. So this is the experimental setting. We wait for 70 days to be able to process the alico slices. Then we seed, because it is a seeding experiment, the exogenous cells, and then we wait additional 30 days to start analyzing the feasibility, the dynamics, the phenotype, the interaction, the function. And it is very exciting to see that the system, the experiment works. So we can see cells in vivo from controls, and they are beautiful. They integrate, they interact, but they don't do much to the organoid. But when we see the cells coming from the patients, we observe quite remarkably an induction of a senescent phenotype in the non-GFP endogenous organoid cells, with a gradient starting from the side of seeding, going moving forward, moving away. So this is the starting point of our future studies. We are very excited, and I'm getting to the end of my talk by reiterating a few key concepts on what Paolo and Mario asked me to deliver today, how to connect MS with other conditions and how to develop an unconventional angle to this connection. I think it's important to keep in mind that mechanisms of chronic smoldering disease are important to study, molecular and cellular. The brain resilience to the disease is a wide concept applied to the number of conditions, which I think is important to be analyzed, including the impact on brain resilience of brain aging at different levels, and the waste clearance system, which I didn't talk about, but they are going wrong in MS, from cellular systems to anatomical systems, including lymphatics and lymphatics. Of course, the responsibility of the next generation clinical trials which are being designed is to assess the reversibility or the druggability or not of each of these key aspects, of course, with the new class of windows of opportunities to stop, restore, or end MS. And this is the end of my talk. I thank you very much for your attention. I would like to thank, of course, the funding bodies, everyone here for the attention, but especially special knowledge goes to FISM and to Mario and to Paola, who have been long-standing supporters and fun and friends above everything. And thanks very much.